Good evening. Welcome to Medicine in Our Backyard. My goodness, we have a full room tonight. I think we have a topic we're all very interested in. This is a series that is presented by the Newport Beach Public Library Foundation. I am Adrienne Windsor. I am a member of the board of the foundation, and so we're very pleased to have all of you with us tonight. Just to prepare for upcoming events, our next Medicine in the Backyard program is February 25th, and it is going to address chronic heartburn. So if you have chronic heartburn, or if you think you might get it, you won't want to miss this. This will be Drs. Kenneth Chang and Brian Smith. And next week, we have another Woody lecture, and that will be on Friday night and Saturday afternoon. Our speaker is Richard Blanco. He was Poet Laureate for the United States. He's going to be speaking on the civic role of poetry for and of the people. Another thing that's upcoming, because I think there are a few grandparents here in the audience, we have a program called Making Memories for Children, and we are featuring STEM experiences in March. And on March 3rd, the Discovery Cube will be coming and doing a presentation. We'd love to have grandparents bring their grandchildren. And then on March 10th, these are Sunday afternoon events, UCI Mind is going to be doing a session on the brain. And it's sort of Brain Awareness Week, and they are going to bring the brain that they have in the lab and gloves, and the children will be able to touch it. So I urge you to bring your grandchildren for that. Now, I'd like to thank our generous sponsors, Mike and Polly Smith. Polly's sitting back there. Polly, will you stand up, please? And Mike, will you stand up, please? And today is Mike's birthday. So can we all wish Mike a happy, happy birthday? We would also like to thank UC Irvine Health for their sponsorship and for bringing this program to us. How many of you here are foundation members? Raise your hands. Oh, wow, this is good. We'd like to see 100% of those hands up. We are a membership-driven organization, and the membership that you pay, the amount of money that you pay, goes to the programs that we have here. Not only our Woody Lecture Series and Library Live, but special programs like Making Memories for Children. We supplement things for the library that the city does not provide. We are responsible for the media lab, if any of you have had the experience of that, computer desks upstairs, the chairs you're sitting on. E-books, so we do many, many things here at the library, and membership is how we do our business. So we appreciate all of you who are, and if you aren't, I hope you'll consider becoming a member. Few reminders. Turn off your cell phones, please, if you haven't done it already. After our speaker, Dr. Federoff, is finished, we will have Q&A, and I will request that you hold any questions that you have until the end of his presentation. So now I have the privilege of introducing our distinguished speaker tonight, Dr. Howard Federoff. Dr. Federoff is professor of neurology at UCI College of Medicine, where he was previously dean of medicine. I can't believe all of the incredible administrative things he's done, in addition to being such a great researcher, because he was also CEO of UCI Health System and Vice Chancellor for Health Affairs. He oversaw the creation of the Susan and Samueli Institute for Integrative Health, the Medical Innovation Institute, and the Center for Precision and Population Health, and he co-founded the Institute for Future Health. Do you ever sleep? His research interests include gene therapy and neurodegenerative diseases such as Parkinson's, Alzheimer's, and prion diseases. He's a nationally renowned clinical and research leader and groundbreaking investigator for neurological disorders. His research led to new gene therapeutic trial in Parkinson's disease that helps predict the onset of Alzheimer's. He earned his PhD in biochemistry and MD at Albert Einstein College of Medicine of Yeshiva University. He completed his residency and fellowships at Mass General Hospital, Harvard Medical School. So we have a very distinguished speaker. Please join me in welcoming Dr. Howard Federoff.
Well, thank you for the warm introduction. Thanks, everyone, for coming out. And I decided today I was going to try to intertwine three themes that really constitute the last 15 years of work that the group has been doing in an intention to provide a level of convergence, because I think we're at a point where there are some new and emerging themes, one of which I'll touch on today, which suggests that the way we had thought about neurodegenerative disease is now turning. And some of the intersections, I think, are going to be important, certainly as we think about the future of therapeutics, but also, I believe, the way we ultimately diagnose some of these diseases before they're symptomatic. So this speaks to itself. And then I decided to quote myself, because I found along the way that individuals who are in training and even some of our colleagues accept something that hasn't yet been proven. And so it leads to this dogmatization of information that actually drives fields in a direction that may not get resolved with regard to really the core of the problem. And so by staying open and asking different, sometimes controversial questions, I think we wind up getting a fuller understanding of the nature of the problem. And I think Alzheimer's represents a good example of that, and I'll illustrate that along the way. So I was thinking about this for the last couple of weeks. So these are the themes that I thought would be good to discuss. What if we knew, predicting Alzheimer's disease before it's symptomatic? When the lights dim, the commonality of energy failure in a variety of neurodegenerative disease settings, and new old clothes, repurposing a drug to turn the lights back on. So what if we knew? So this is a frequently used drawing of an individual. The motif is the whole foliage, as well as the individual on the right, who's now losing part of what constituted their normal cognitive function. I think it's interesting. I often will show a couple of pictures, not for this evening, about artists who themselves were beginning to enter into dementia. And their artwork, particularly self-portraits, the way they began to reimagine the way they looked, which was quite different than they did when they were preclinical. But this is, I think, a good beginning. So many people will use a slide like this to talk about Alzheimer's disease, right? Dr. Alzheimer's himself was trained as a neurologist and a neuropathologist and took care of the patient on the right who was admitted to a local hospital after having a failure to thrive and some cognitive problems. Everyone knows that AD is associated with loss of brain volume, but I want to use this slide not only to refer you to the most common initial presentation, which is loss of memory function, and then ultimately change of life and quality of life, but I want to talk about amyloid. This is the dirty plaque that occurs in the brain. And you heard earlier about Brain Awareness Week. There's a lot of things that have been said about amyloid, and it's been so much in the forefront of research because it was defined as a neuropathological, when you look at the tissue of someone who in life had a clinical diagnosis of dementia, most commonly Alzheimer's, and therefore the equation between this as being the offending causative contributor to Alzheimer's disease had really spawned the hypothesis, now called the amyloid hypothesis, almost 30 years ago, when Dennis Selko and John Hardy put this forward. The problem with this is that we've now done many, many hundreds of clinical trials, and all of them that have targeted amyloid have failed. In fact, most interestingly and disappointingly is that you could demonstrate a reduction in amyloid burden in some of these clinical trials, so it goes down, yet people don't get any better. So this really is, when I said the world is beginning to change, I think the last 3 or 4 years have made clear that there are other thoughts about what might be going on, and I'm just going to share with you one perspective. So we began this work more than a decade ago, and we decided to take a different approach. While we did not know then what we do now about the failed clinical trials in amyloid and clearing it, we decided that 2 things were important. One is that this had to be a systemic disease, irrespective of the fact that it presents clinically referable to changes in the brain and some of the processes that are subserved by those brain networks, principally cognition. We knew it had to be systemic because there were other things that had been hinting at this for some years. And so the question is, if we could find a systemic feature for Alzheimer's disease, could we find it early and maybe even preclinically? And that's what drove our research early on. And so we designed a relatively simple study. Actually, I started this work when I lived in Western New York, where actually today it's very cold. But I was gratified in that I had been coming to Irvine pretty much every year to visit the Alzheimer's Disease Research Center when Carl Kottman and later Frank Laferla were running the ADRC as an external advisor. And I met Claudia Kawas. I actually met her a long time ago, but she became our local collaborator for what we now call the Rochester Orange County Aging Study. But the idea was simple. We would enroll seniors, and we would just follow them clinically, neurologically, and neurocognitively. And some of them, unfortunately, developed cognitive impairment that we would document. And at the time they developed this, we went back to the samples that we had collected from their blood at the time of entry when they were cognitively normal. And we asked, is there anything in the blood at that time that would have predicted that that individual or that group of individuals would go on to develop cognitive impairment, mild cognitive impairment, or Alzheimer's? And so the nomenclature that we adopted is that these individuals who came in cognitively normal, they were destined to convert, phenoconvert, to being cognitively impaired. And so we labeled them converters, but before, pre. And then after they converted, we called them converter post. And a more familiar nomenclature was that these were the individuals at risk, and these were the individuals who already carried a diagnosis. We've done a lot of things in this cohort, but the only piece of that, which I'll share with you today, is the work that was focused on blood metabolites. But we've looked at every informational molecule that you can in this group of 525 people. And the way we went about doing this is we enrolled individuals, and they had to be cognitively normal. So we went to family medicine, internal medicine practices, we went to competing health systems, and we said, we're doing a longitudinal study, and can you refer in individuals who are cognitively normal from your practices? Unfortunately, some who did get referred in actually had newly diagnosed MCI or Alzheimer's disease. And others were normal, and we divided the work that occurred over a period of 5 years into what we call the discovery phase, and then subsequently the validation phase. We had hoped that in the discovery phase, by looking at something in the blood, and as I mentioned, I'll just share with you the data on the blood metabolites in that fraction of blood, which is called plasma, we had wanted to be able to develop a robust, highly sensitive, specific, and accurate way of classifying these individuals. And we wanted to do that when we were agnostic in terms of where that blood came from. So the classification power was really requiring for this to be trained in discovery and then validated subsequently as we recruited additional people. So we think of this, although this is an image intended to convey probably more than we can do, we thought that there would be mirrored reflections of what was going on within the brain, in the blood. And when we started, we really didn't know what it is that we would find. And there's been a lot of unanticipated and interesting observations, including the one that I'm sharing with you now. And what we found was that there were a group of small chemicals. Most of these are actually lipids, fats. And some of them are a little bit more complex than others. And almost all of them were lower in individuals who were at risk, the converter pre's. And then as they progressed through MCI and AD, they came back up nearly to normal. So it's an interesting observation. There had been lots of work that was done historically, not by us, but by many other groups, looking at lipid changes in the brain of Alzheimer's patient. And there were others who had really plowed some of these ideas before we got involved about a decade ago. But what we ultimately wound up describing, and here I've collapsed the data because we're measuring concurrently 24 different analytes, these metabolites, into what I'm calling an index, because we had envisaged that this would be used clinically, and we wanted to have a way to collapse the data so that we could make the distinction between someone who's at risk and someone who is not. And so these are the normal controls, and these are the individuals who go on to develop Alzheimer's disease, or MCI. They're non-overlapping. And it took a lot of work informatically and otherwise, but we basically have developed this blood-based measure that is approximately 90%, 96% accurate. There's a lot of interesting things that one could talk about. I'm sure some will surface in the Q&A, but let me just continue. So we were really interested in how to interpret this. And so I'm just going to share with you a couple of things, and I apologize for showing you a biochemical pathway. But Eugene Kennedy in the late 50s, working in Boston, had described the basis upon which we generate phospholipids. Principally, the two are phospholipids based on the choline backbone or the serine backbone. And so this is a pathway that he had been studying and had been validated in many, many different species, including in people. And when we reported our first result, what we were unaware of was that there was a subsidiary of Dannon Yogurt that was headquartered in Amsterdam that had developed a medical food with a supplement that was based on the brilliant work of Richard Workman, who now is emeritus at MIT. Like myself, he and I both trained at the MGH. He stayed at MIT, and he pioneered the identification of the nutritional biochemical requirements to build a synapse, the connection, and to have it sustain its function. And what Dannon did, Nutricica, is they took a group of naive, meaning that they haven't seen a drug or they haven't seen this product, which I'll tell you about in a moment, and I'm not endorsing this product, and I don't consume it myself. And they treated them with this particular supplement as part of a medical food. That's how it's registered in the European Union. And this is the composition of it. These are the key things that came from the Dick Workman's work. So one of them is uridine monophosphate, and you need uridine in order to be able to build several of the components that ultimately lead to CTP. These are omega-3 fatty acids, choline, phospholipids, including some that I'll show you in a moment, which were changed in the work that we had done, folic acid, vitamins, and selenium, because some of the enzymes require selenium as a cofactor. And then they went back to their freezer, because they had been doing this work, unrelated to our own. And shortly after our first volley of work was published, I get a call from the lead investigator, and he says, "We're really interested in your work, and we have a paper coming out next week." I said, "Okay." They couldn't measure everything that we did for a variety of reasons, but what I'm showing you, and you can ignore the chemical nature of what's on the left, but each of those that were asterisked here in red was something that we defined as being low in people who would develop MCI, mild cognitive impairment, or Alzheimer's, that on this product, they had restored to normal or above normal. So the Wortman legacy was defining the basis to build, sustain the function of a synapse. This ingredient cocktail was what he had worked on, licensed to Dannon, and they built this drink, which I'll now tell you is called Souvenir. And this is how they had started down the path to do nutritional supplementation with an intention to restore or stabilize synapses. The jury's out, but I think the work speaks for itself, and it really is, as I said, a testament to Dick Workman's 35 years to really map this out. The second insight from our work was that there's something about those lipids that are changed in the blood that told us that it was highly likely that there was something failing in terms of energy production in the brain. The energy factory of the cell, it's an organelle called the mitochondrion. It does a lot of wonderful things, but it can decrease in number per cell, or it can become compromised in its function, and it causes the cells to have effectively energy depletion. So the currency of energy is this high energy triphosphate, ATP. It's made from both glucose, but it can also be made by burning fatty acids, lipids. And it does so in a way that's highly regulated, and some of what we have found changed when you're at risk for Alzheimer's disease is that some of the lipids that need to be burned and others that need to be transferred in order to be burned were downregulated. So this observation allowed us to begin to think a little bit more about this mitochondrial process. My apologies for this, but I'm just going to tell you that this nice little blue blob is an acyl-carnitine synthase. And it is required in order to be able to transfer those long-chain fatty acids, shown here, which we found reduced in the blood, into the mitochondria in order to be able to be burned and to make ATP. I could go on about this because there's a lot more insights, but I'm just going to leave this one here and happy to respond if anyone has further questions later on. So just to summarize this piece, what if we knew? Well, we now have a highly accurate blood test that can predict the development of Alzheimer's in seniors. We don't know how young. Everyone we enrolled were over 70. These blood fats that I was describing are decreased. And one interpretation is that they are being shunted into the brain, perhaps in an attempt to maintain synapses. We don't know that for sure, but that's our leading hypothesis. And others of the blood fats that I described are decreased, suggesting that mirrored in the blood is this energetic problem in the brain. And I'll develop that a little bit further in the next section. So when the lights dim, this is the commonality of energetic failure. And this didn't really occur to us until about 4 or 5 years ago. But you know, we often think that it's an all or none phenomenon, but actually it's very graded in terms of the bioenergetics of a cell. But there are certain things that can happen where you have nearly a complete decrease in the ability to sustain the requirement to make energy. And although the switch, as shown here in the cartoon, is not truly fully off, but it's very much close to being off. And I'll remind you that the human operating system, which requires the energy molecule ATP, is powered by the mitochondria. No mitochondria, no ATP. It's a big problem in sustaining function. So we came to this from our work in Parkinson's disease, but I'll generalize it in a moment. We were interested in a problem like many, which I'll describe in some detail, but basically this is a sagittal section of the brain showing the parts of the brain that are affected in Parkinson's. This part in so-called midbrain, the substantia nigra, is where the cells have their so-called cell bodies that make dopamine, and they send their processes up to this part of the brain, which is often called the striatum. So loss of dopamine neurons, loss of dopamine is equated with some of the clinical features of Parkinsonism, which include slowness of movement, usually tremor. Later on, there's difficulty with gait and many others. But we were interested in the problem since it had been described like 3 decades before we got into this, that Parkinson's patients, if you looked in their platelets, they already had evidence that their mitochondria were impaired, but no one knew why. These enzymes that are part of the respiratory chain that make ATP were shown to be down in platelets, and later it was shown in brain tissue from individuals who unfortunately succumbed to their Parkinson's disease. And so we and a group of other investigators were trying to get to, well, what's going on? Why is it declining? So 14 labs across the globe collaborated, which at the time was a pretty unprecedented thing, and I really congratulate the person who first put this on the map, Clemens Scherzer, who's at the Brigham and Women's Hospital. He's a movement disorders neurologist. And we all agreed that we were going to share all of our data, and we worked with the MIT Broad Institute, who had been developing advanced informatic tools. Not only did we confirm and extend the finding that the respiratory chain genes, these are the things that are responsible for making ATP, were in fact down, but we did for the first time find that the master regulator of mitochondrial function, which has this fancy name, PGC1 alpha, was decreased in Parkinson's preclinically, but it stayed down throughout the disease. So this was a new observation, and one that I think warranted a lot more work, and we and others are following up on it, and I'll share some of that with you. So this cartoon is intending to tell you a few things, but normally this gene, which can be expressed in a variety of tissues, in fact, it's nearly ubiquitous because it's so important in regulating energy throughout the body. It has a basal level of expression, but it could be brought up and it could be brought down. Interestingly, and this is a connection, and now this is being now evidence-based in the treatment of Parkinson's patients, exercise of moderate to high intensity is able to increase the expression of PGC1 alpha in skeletal muscle and other tissues. And that's why many patients are now being recommended that as part of their overall strategy to slow the progression of the disease, that exercise of a structured type needs to be done. What we know is that this works through a number of different pathways, but the goal ultimately is to make more of these, thus the mitochondria. And so you can just imagine for today that if you make more PGC1 alpha, you get more mitochondria. If you make less, you get fewer mitochondria. So this slide is to tell you a few things. I'll start off with the green boxes. So the consortium that the 14 labs, we found that these gene names, which are all shown in green, were all decreased in preclinical Parkinson's throughout the course of the disease. And I'll tell you what they mean in a second. sort of at a meta level. They're part of these different respiratory complexes, complex 1, complex 2, and complex 5, which are part of what the mitochondria does in order to generate ATP. So multiple genes were being coordinately downregulated. And parenthetically, and not so surprisingly since I've already told you, many of them are regulated directly by PGC1 alpha and some indirectly. So by going to the apex of this pyramid, if you will, and finding for the first time that PGC1 alpha was down even before patients had a unilateral tremor and then stayed down, provided the basis to start thinking about this as a new drug target, which will be the last subject I'm going to cover. So I'm going to spare you a lot of experimental detail, but we've shown that what turns this down, including in a variety of different models, and we've also demonstrated this in human postmortem Parkinson's tissue, is brain inflammation. And interestingly, there is at least one very potent free fatty acid that is able to turn this gene down. I told you a lot about lipids. Fatty acids are lipids. We know that the mechanism through which both brain inflammation, usually mediated by a variety of so-called cytokines, and as well the saturated fatty acid, they don't change the nature of the code that encodes PGC1 alpha. What they do is they prevent the gene from being expressed normally. And they do so by a very specific chemical modification of the beginning of the gene that is essential for it to be expressed. That's the epigenetic change. So this means that if PGC1 alpha expression is epigenetically modified, there may be the basis upon which we can restore it. And the question is, how could we do that? So summarizing a lot of data, some ours and some other investigators, if chronic inflammation is causative to decreasing this PGC1 alpha, is it also affected in other neurodegenerative diseases? So it is indeed, at the time of diagnosis in human cortex, it's downregulated and it goes down further with progression. It is downregulated in amyotrophic lateral sclerosis, Lou Gehrig's disease, both in the part of the brain where the upper motor neurons live, as well as in the spinal cord. Interestingly, in the very marked Mendelian neurodegenerative disease, Huntington's disease, it's down. And recently, working with Dr. Chris Kenny at the Gavin Herbert Eye Institute, we've demonstrated that it's down in age-related macular degeneration, something that wasn't even on the radar 18 months ago. So new old clothes and repurposing a drug with the hope to turn the lights back on. I just remind you that it's about ATP generation in mitochondria. Drug repositioning has been around for a very long time. It simply means that you are looking at an FDA-approved drug and asking in a different clinical indication than the FDA had approved for its initial registration, whether your drug might produce benefit in that other indication. And there are myriad examples. And although it may seem like it's a simple question, it often is far more complicated, and I'll show you some of the nuance in a moment. Why does one do this? Well, if you're like me, you're impatient. And so if you look at plotting risk, cost, and time versus return, you can see that repurposing occupies this quadrant because it gets done more quickly, it costs a lot less, and its success rate is nearly 10 times higher than taking a new chemical, which is often called an NCE, a novel chemical element, and prosecuting that through discovery and clinical trials. And those are the proprietary drugs that often, rarely, but often when they are released, they actually are very costly. So the other rationale for going this route is we really wanted to take a value-based approach, both in terms of time to, but also the cost of a per unit dose. So we screened thousands of FDA-approved drugs. We did a 2-tiered screen in one cell line, which is a retinal cell line. We identified 14. We corroborated all 14 in a variety of other cellular systems. And we selected this one, which we call a PGC-1-alpha upregulator. This was the 91st one in the array that we had screened. And we selected it in part because it's been prescribed all over the globe. It's no longer proprietary. And it's been estimated to have approximately 6 million patient years of experience, and it's safe and well tolerated. So if I do a study in a mouse and I give a very high dose of this PU91, I can detect it in the liver and I can detect it in the brain. But it takes a very high dose, approximately 10 times higher than you would administer to a patient for the primary indication for which the drug was initially approved. And I'm going to show you some evidence which suggests that it's protective, because you could administer after you start neurodegeneration in a Parkinson's model that we do in the mouse. This is probably the best mouse model. I'll just cut to the chase here and tell you that you can protect the brain against inflammation by starting the treatment after neurodegeneration has already occurred. So it's anti-inflammatory. We also measured its ability to save nerve cells and save the terminals. Remember, I talked about the terminals that are in the striatum that release dopamine, as well as the cell bodies which lie in the midbrain, remember the substantia nigra. And we can do both. We can save nerve cells and we can save nerve terminals. So a very, very high dose. So we were very excited by this because we thought it was going to be a very clean path now to move into clinical trials. But the dose was wrong, and then this is the problem. The FDA approved a prodrug that, when metabolized, is the active drug that the FDA approved for its original clinical indication. We needed to know which one of these was responsible for the effect that we were seeing. And as it turns out, the prodrug, which was thought to be inert, the thing that the FDA thought was just the vehicle to get the active drug, its metabolite, approved, was the thing that was turning up PGC-1-alpha. We've done a lot of work on this, and the active drug for the primary indication is completely inert. It does not work to turn up PGC-1-alpha. So it was the prodrug that we had to focus on. So we did a fairly conventional study by looking at a single dose to see how rapidly it's metabolized. I'll point out here, we gave PU91, and we measured its concentration in liver and in the brain, the substantia nigra. And look at the scale here, 4 and 6. These are nanograms per mil. If you look at the metabolite, now the scale is broken because it's 1,000 times higher concentration of the metabolite, which means that the body, principally the liver, is seeing the prodrug, and through principally a single sweep through the liver, converts 99.9% of the prodrug into the metabolite, which is not satisfactory if you want to get the prodrug into the brain. So we had a new problem, and I won't go through all of the details, but we identified the human enzyme in the liver that's responsible. And then we relied a little bit on what I'll call biochemical intuition, computational chemistry. And we identified a natural product which is able to inhibit, not completely, but inhibit the conversion of the prodrug to its metabolite. So what I'll leave you with now, it's still a work in progress. You don't want to ask this person to take a look under the hood. But what we are moving to do, in fact, it's actively involved in evolution, is to co-formulate them so that they can be delivered together. But I will tell you that they have to be delivered in a precise ratio relative to one another, because otherwise you don't get the effect to upregulate PGC1 alpha. So here's one experiment in the right ratio. When you administer them together, again, it's a simple experiment. It's a one dosing. You get a 4 times increase in the amount of the prodrug into the brain when you get the right mixture. Interestingly, you also get an elevation of the metabolite, not so surprisingly in the brain, because once the prodrug gets there, there's a little bit of conversion in the brain of the prodrug into the metabolite, even in the brain, which obviously isn't the liver. So we now have a method to go forward. So let me just try to bring this back into frame. I already told you about this master mitochondrial regulator that can be turned up by exercise. It can be expressed basally, but we've now defined an FDA-approved drug that is able to turn it back on. And actually, we think we know the precise proximal mechanism of this right now. And we can demonstrate, and in fact, I alluded to the work that we did with Chris Kenny at the Gavin Herbert Eye Institute. We not only know, at least in her models of AMD, that we can restore it, but it has all this downstream benefit. You get mitochondrial function coming back to normal. You get inflammation inhibited. You get cells protected. This is mirroring all the other work that we've done outside of the eye. So this is a common observation that is suggesting, as I said at the beginning, pointing to convergence. There's a common mechanism that seems to undergird multiple degenerative diseases, whether they be in the neuroretina or in the brain and the spinal cord, as is in the case with ALS. So what are the conditions? Well, I've already mentioned the work that Dr. Kenny is doing. This is really spectacularly interesting work. We believe, given where this co-formulation is and the nature of the Alzheimer's disease models, which are not, they're all amyloid models, that we could move a little bit more expeditiously into clinical trials if we could demonstrate safety and tolerability. But recently, the Department of Defense has become very interested in this, and they've invited us to apply to receive funding. Of course, you have to still go through the formal process, but to see whether this might be a safe adjunctive measure after someone has had traumatic brain injury of a mild or moderate type, where you know it's going to be safe and well tolerated because it's already been exposed, patients have been exposed to this. So we're doing this work with Dr. Brian Cummings just as we speak. So traditional drug discovery, which you're familiar with, takes a long time. It has a high failure rate. You saw 2% success rate. Repositioning, we're right over here. We're almost done with preclinical development. We're trying to secure the rest of the funding that we need to go forward to get our first so-called investigational new drug application so we can start the work in patients, the IND, and it will occur at a fraction of the cost. I can tell you that what we don't talk about is how many grants we've all received. So this has been governmental funding and some philanthropy, but we've been able to, over a decade, be able to bring this into what I think now is a nice frame. And as I hope I've left you with, a strong sense that this commonality may wind up making a therapy like this beneficial in more than a single clinical indication. So we formed with Frank LaFerla and a business partner with the intention to accelerate this work, a little startup here in Orange County. And now I'm just going to summarize in 2 different slides. First, the new old close, the repurposing story. We've identified a new target for neurodegenerative diseases affecting both the brain and the eye. We can restore the levels of PGC1 alpha, and we understand how PU91 works. In addition to increasing the levels of PGC1 alpha, it blocks inflammation. It also blocks reactive oxygen species generation because it lies at this apex and is able to promote the activation of multiple beneficial genes, which almost is exactly what one needs in a neurodegenerative disease state where you have many, many different things going on: inflammation, oxidative stress, cell loss, synaptic dysfunction and failure, and mitochondrial failure. And we are able to increase this in the brain by the co-administration of a natural product that you and I have all consumed. But I'm now having this product made to a very high degree of purity so that I can work with the regulators to have this be something that can be co-formulated with the PU91. So if there are take-home points, let me just leave you with blood lipids are altered in preclinical Alzheimer's. We have a highly accurate blood test, about 96% accurate. These blood lipids suggest that there's an energy deficit. There is a commonality across a range of neurodegenerative diseases regarding the decrease in the PGC1 alpha. We have turned our attention to repurposing a drug to turn it back on. We've identified the candidate, and now we understand how to make more of this available in the brain by the co-formulation, co-delivery of a natural product. The work always takes a lot of people, and so over the years I've really been blessed to have both good funding and great collaborators. Both when I started this work at Rochester, I continued at Georgetown, and the team in part moved out here to UCI. I have also worked closely with several individuals at the NIH, Mike Knowles and Andy Singleton. And so with that, I will give you all a big thank you. Very few of you fell asleep.
Thank you so much, Dr.
Sure,
Federoff.
sure.
It's so lucid, so wonderfully presented, and so inspiring. Now, Dr. Federoff, we'll take questions from you. We have so many people here tonight. I'm not going out into the audience with the microphone, so I'm going to ask if when you ask your question, you will stand, and then if you would repeat the question so that the audience can hear it. So I'll let you choose your questioners.
Please.
Just an easy question to begin.
So you have the hard ones coming up next?
Dr. Brian Cummings, what facility is he with?
He's at UCI, yes. He's in the Stem Cell Institute. Yeah. Please.
I'd like to know what is the blood test, and also how did you, were the brain samples on the graphs that you mentioned, was that from a mouse model or from a human?
I'm not sure which one we were talking about, but let me come to your first question, then I'll ask you some more to get to the answer to the second. So all of the blood work that we've done are in humans. I can tell you that actually we've replicated some of this work in animal models. And the other thing I can tell you, is, which is very surprising, is that the rare form of Alzheimer's, familial AD, where you get the gene and you have the disease, some of the same features that we discovered are altered there, suggesting that if you have this mirroring effect, it's independent of whether you have the more common late onset form of Alzheimer's versus the very rare familial, which is something we would have never expected. So it's a general feature, and I use mirroring because I can't yet put my finger on exactly the nature of the relationship. Now, you asked me about animals. What?
No, like you mentioned that you have one blood test that is 96%.
Yes, that's in people. That's in people. Yeah.
Okay. And when you showed the charts, you showed that there was a level, different levels noted in the, what was it, the striatum?
Yeah, those are animal studies. that have an animal model of Parkinson's disease. We have not tested the combination yet in any human volunteers. That's next. We have to go through the late part of the regulatory process before they'll grant us the so-called IND, but we're anxious to do that soon. Yes, back there.
Where would any of us living in this area go for this test? What test do we ask for?
Yep. So it's not yet commercially available. That's a problem. I have been interacting with a lot of large diagnostic companies. They are very much interested in staying with the amyloid hypothesis. So there are tests that have been reported on. None of those are yet experimentally or commercially available. What I've said to Frank LaFerla, who's the head of the ADRC, if patients who are seen as part of that longitudinal study or even cross-sectional study, I would be happy to be able to offer this on an investigational basis. It's not going to be paid for by insurance, so there has to be a way that the ADRC could pay for it, but that's how I would do it currently.
Quick follow-up. And what role, you mentioned it, but it's not clear. What role is genetics in all this? Parents, grandparents, strong case that you
No.
had or not?
No, so let me repeat
Please
the
repeat
question.
the question.
So the first part, everyone heard, where could you get it? And I commented, and there may be a path forward with regard to the UCI ADRC. The second question was related to what role does genetics play in this? Right now, since I just shared with you something that has not yet been published, that some of these same measurements, whether you have genetic form of AD or the much more common late onset, they seem to all be very, very similar. So the answer to your question probably is it's not going to play a big role. And it really does suggest that the measure is independent of the cause, right? It's telling you about cognitive function. And I'll tell you one thing that I left out of this talk because of time. But in our cohort of 525 people, we had individuals who not only were normal, their neurocognitive function was supernormal. And we could define 12 different measures in their blood that can classify supernormals from normals, from patients who have cognitive impairment. Starting with the fact that there is a supernormal signature, it suggests very strongly to me, because it's not a disease, that classifier is telling you something about the brain's cognitive function, whether it's neural circuits of one type or another, I can't say yet. But I'm now beginning to believe that irrespective of what might be upstream and etiologic, we have now defined a basis of something that is mirrored in the blood that I believe generally reflects some cognitive change. Please, over here.
The 96% accuracy of
Yeah.
the test, very impressive. What is the false alarm rate?
Yeah, so we've done sensitivity and specificity. So sensitivity is nearly 100%, and specificity is a little bit lower than that. What we've recently done, this is also unpublished, is that we now have externally validated this, because I was a stickler for details. I left this part out. Everyone who was part of our original study had to come into the clinic in the morning. They had to withhold breakfast. They had to withhold their morning meds because we wanted to control for circadian rhythm. We externally validate this on patients that were not part of our study, who were seen any time during the clock time, and we're still getting an accuracy that's about 93%. So my sense is that the accuracy is going to depend on the standardization of this. And so as I think about the rolling out of this, and what my intention, if I'm able to get this done, is I'd love this to be really low cost. It's certainly non-invasive relative to some of the other things that are done, and to become part of CMS's annual wellness visit, because Alzheimer's has become a national strategy, and I'd like to do this. Remember, I said value. I want this to be done at a very low unit cost per test, because I think we owe it to ourselves and I think ultimately, you may be aware of this, but in fact, my collaborators published on this, but there is data that suggests that knowing early actually causes an enormous amount of health savings cost and also an opportunity to engage in other activities that now are beginning to yield some evidence that they're slowing progression. So I think there's a pretty compelling logic to doing it then, part of the CMS annual wellness exam. And so my mandate is really wanting to move this so that, notwithstanding the fact it has nothing to do with amyloid, has nothing to do with genetics, as far as we can tell, we want to move this so that it could become a tool that clinicians in the community can use in helping to decide how to best inform their patients and their families. Please, over here.
I have a question about the drink that was.
Yeah, Souvenir. Yeah.
I kind of wondered why, or if you thought highly of that, and what's the repercussion of taking it that you decided not to have it? And does that have any relationship to the product that you were developing? Does, you said you're developing something but with a high level of purity. Is that a medicine or is it a?
Yeah. So I don't.
Can
I
you
don't
please repeat the
Yeah.
question?
So the question was about the Dannon subsidiary Nutritious Souvenir, which contains the ingredient list I showed you that was worked out by Dr. Richard Wortman at MIT. I don't have any reason to endorse it or not. The clinical data about improving is just not there. So if I looked at this purely from a clinical perspective, I would say there's no yet justification to recommend. So what I elect to do for myself may not be what I would elect to do or recommend to a patient. With regard to our natural product, it has nothing to do with anything that Dr. Workman's group had defined. And we picked that natural product largely because it inhibited the conversion that I described of the prodrug into the metabolite, and we need the prodrug in the brain. But it has one other benefit, which I haven't told you, is that the natural product is a very good anti-inflammatory. So it is an additional benefit. And we didn't pick it for that reason, but that's a sort of an added benefit. So our strategy going forward is to develop this combination product. We've talked with a former regulator who used to run neurology for FDA. I think we have a bit of a roadmap. And it's really a question of how do we get there quickly, and in which clinical indication do we go for first, since there seems to be this commonality. Please.
Does a blood thinner have any effect on the blood test?
Not that we know of. We've not looked at we've looked at some of the newer antiplatelet drugs. We certainly have looked at Coumadin and heparin. We don't see any impact. So our sense is no, but we haven't done it systematically by looking at groups of individuals who are on this drug versus that drug versus another, and then do the test. So I can't really definitively answer that. There's a question here in the back. Please, in blue.
Would you like to identify the natural product?
Not now. No. No. It doesn't come from fish. This is like 20 questions now. No, it's not yogurt. It's a product that's made by a subsidiary of Dannon called Nutritious. It's headquartered in Amsterdam, and they produce, under parlance of the EU, a medical food which contains the supplement that it's a set of ingredients. That's Souvenir. So if you Googled it, you actually could buy it at Amazon, and I'm not suggesting that you do, and I have no interest in Dannon or other, and as I've already mentioned, I'm not recommending it. But if you had more interest, that's how you could find it. Over here, please.
Yes, I wondered, what was the name of the LLC?
Yeah, so Frank LaFerla, as you know, he's of Sicilian origin, and he really wanted to name it Menta. And so it's of Italian origin. And so Menta Biosciences is what we call the LLC, and Frank and I co-founded the company just about a year ago. Please.
Can you speak for a second about any interaction that you've had with the National ALS Foundation or other ALS-related organizations with regard to the implications of this research for ALS?
Yeah, great question. We haven't had any yet. I would say
Please repeat it.
Yeah, I'm sorry. So the question was, have we interacted with any of the national foundations that are interested in finding a cure for ALS? And the short answer is no, not yet. Please.
Are your findings relevant to frontal temporal?
Yeah, we haven't done FTD yet, but it has some of the signature that we probably would imagine. There's chronic inflammation. As you probably know, there are multiple forms of FTD, ranging from that is multigenic or polygenic to those that actually carry a single Mendelianly inherited mutated open reading frame. We think that probably has some of the same features, but we just haven't gotten to make those measurements yet, so it's a great question. Please.
So would you say that those are still investigating amyloid are misguided?
I'm sorry, what?
Those that are still investigating amyloid is misguided.
I probably wouldn't use those words. Yeah. So what I would say is that the amyloid hypothesis has been very powerful. It's undeniable that amyloid accumulates in the brain. Actually, Rudy Tanzi, who's a well-known AD investigator, now believes that amyloid is there to actually promote benefit as opposed to causing harm. We think it's a neuropathological signpost. We think there is some evidence, particularly when you have large amounts of amyloid, that you can trigger things. But we believe that in the AD brain, it's not driving disease because it's undeniable that when you even remove it, no one gets better. And those who are still, the proponents of it believe that you have to go earlier. And I say somewhat tongue-in-cheek, are you going to treat a 10-year-old with an anti-amyloid drug in the hope that you'll be able to prevent it? So I think the jury's out. There's one set of clinical trials that are getting done now, being sponsored by Biogen, in patients who have familial, which is the pure form of amyloidopathy, because you have a mutation in a gene that causes more amyloid. If it doesn't work there, I think it's game over. I'm not buying the pediatric hypothesis, even though I mentioned it tongue-in-cheek. Please.
Are you optimistic that if you get this drug perfected with the combination, that it would be useful in early use, pre-use, or would it also be useful in treatment of more developed patients?
Yeah, great. So the question was, will this combination be useful both early and maybe later? And we don't know the answer to that. Our suspicion is twofold. One, that something that might be disease-modifying is likely to have the greatest potential to be disease-modifying when administered early. MCI patients already have a substantial amount of disease. I would not rule out, because they have not yet gone to full-blown Alzheimer's, that it would not have any benefit. There's a lot of road ahead of us here, so I'm just wildly speculating, but my sense is that the FDA will have its own thoughts about in whom we should try this first, and it's likely to be patients who carry a diagnosis. That'd be my guess. Yes, in the back.
One more question.
I was just curious if the word inflammation is carried throughout on your
Yeah.
talk tonight. Has anybody doing any kind of parallel studies in dietary changes that would just bring about a reduction of inflammation?
Yes.
Cardiac or brain? Or
Yeah, great question. So
Please repeat it.
we mentioned inflammation. Actually, systemic inflammation is a frequently described component of many diseases, including those that don't affect the brain. It affects the vascular endothelium, both the macrovascular, like in the upper extremity, lower, the microvascular, which could include the penetrating non-epicardial vessels of the heart, as well as the brain. So yes, there is a lot of interest in wanting to reduce inflammation. And the question is, how does one do that? Because some of those very potent anti-inflammatory drugs put people at risk for infection. So for example, the whole class of drugs that are TNF antagonists, there's a whole set of biologics, Intercept is one, but they basically dampen down the major action of TNF-alpha, which is etiologic in those diseases, but it puts them at risk for fungal infections, tuberculosis, and others. And so I think that the next generation of smart anti-inflammatories are likely to be not just a single drug, but probably others that would be co-administered so that they don't produce the risk of what could be a life-threatening infection, but are really targeting some of the inflammatory cascades without necessarily putting at risk the individual for some type of infection. But I think, yes, you're spot on. Thank you, everyone.